Hello, everybody. My name is Tzion Fahima, and I'm coming from the University of Haifa in Israel. Today, I will talk about the discovery of the Tandem Kinase-Pseudokinase protein family and the proposed decoy model for their involvement in plant immunity. Striped rust is one of the most devastating diseases of wheat. 88% of wheat production in the world is susceptible to yellow rust. It is causing losses of 5 million tons per year globally, worth of $1 billion. The best way to fight yellow rust is by using resistant cultivars. WIRE15 is a yellow rust resistant gene conferring a broad spectrum resistance to yellow rust. It showed resistance against 3,000 isolates from 47 countries around the globe. Here on the right, you can see neurogenic lines, avocet susceptible and avocet plus WIRE15. Avocet susceptible is showing impressive symptoms of yellow rust, while the avocet WIRE15 is showing hypersensitive response type of resistance. In collaboration with Morgan Hovmoller in Denmark, we have shown that WIRE15 is activating a programmed cell death response, a hypersensitive response. The invaded mesophyll cells are collapsed and distorted, and you can see the aggregation of chloroplast, and you can also see the autofluorescence, which is indicative of a hypersensitive response. On the left, you can see the susceptible avocet. The fungal biomass is accumulated, the pathogen accumulated fungal biomass, and there are no indications for a hypersensitive response. WIRE15 was cloned in 2018, and using the map-based cloning approach, I presented the results in the previous PGRI workshop in Marrakesh, and the paper was published in Nature Communication. And today I will show you what we have done since the cloning of the gene. WIRE15 is coding for a Tandem Kinase-Pseudokinase protein, TKP, designated as wheat Tandem Kinase 1. It has a unique structure of two kinases in tandem. One is a kinase and one is a pseudo kinase. The size of the gene is 665 amino acids. We have searched the clone gene from Dacocorides. We have searched to see if reference genome of bread wheat and wild emmer wheat, Zavitan, if they contain the gene, because both of them are susceptible. We wanted to know if there is a non-functional allele of the gene. And indeed, we found non-functional alleles of the gene, both in Chinese spring and in Zavitan. We wanted to know also why they are susceptible. And what we found is that transposable elements were inserted into the genes and disrupted their activity. We wanted to know if there are other, how many copies are there in wheat genome, bread wheat genome, and if other diploid and tetraploid wheats contain also copies of the gene. And indeed, we found more than 20 copies in bread wheat. We found orthologous copies on group 1 chromosomes, on 1A, 1B, 1D, and paralogs on 6A, 6B, 6D. We used the information to develop functional molecular markers for WIRE15. You can see here, we use neurogenic lines. On the left, these are the resistant lines with WIRE15. On the right are the susceptible lines without WIRE15. And we tested them with these functional KASP markers. And you can see that all the resistant lines show the resistant allele of WTK1, while all the susceptible lines show the non-functional allele of WTK1. Then we wanted to know if there are other functional genes that contain this structure of tandem kinase structure. And indeed, the RPG1 barley stem rust resistant gene has this structure. Also barley through loose mud UN8. And recently, George Dubkovsky cloned the SR60, WTK, he named it WTK2. And Tzion Liu in Beijing, he cloned powdery mildew-resistant gene PM24. All of those genes contain this structure of tandem kinase gene and belong to this family. Then we wanted to know how common are these genes in the plant kingdom. And we searched 11 genomes, 11 plant genomes, and we identified 92 TKPs. And we compared them to the Arabidopsis kinome. And this is the phylogeny analysis, and you can see that they belong to genes that are involved in kinases that are involved in immune responses in Arabidopsis, also in rice. Genes like the wall-associated kinases and the cysteine-rich kinases. When you look at those genes that are functional, the 5 functional genes, you can see that some of them evolved by gene duplication events, like WTK3 evolved by a duplication of cysteine-rich kinase. Same is true for RPG1, while others like WIR15 evolved by a fusion of 2 different kinases, the wall-associated kinase and RLCK. So the question is, what is the role of the tandem kinase-pseudokinase proteins in plant defense? And we came up with the model. We proposed the decoy role for the pseudokinase domain as one of the potential mechanisms of function of the TKP family in members in immune responses. What we can see is the pathogen stripast is invading into mesophyll cells. Through the haustorium, it is excreting effectors which are suppressing the resistance response of the plant, the immune response. They are interacting with receptor-like kinases and suppressing their activity, and the result is that the resistance cascade is blocked and the plant is susceptible. While during the evolution, what happened in the plants, that they evolved this TKP family, which is a duplication of the kinase or a fusion of 2 different kinases, while the effector of the pathogen is trying to switch off the defense response controlled by these receptor-like kinases. They actually switch on when the effector is interacting with the pseudokinase. It is actually activating the kinase and activating a resistance cascade, leading to program cell death. Conclusions: the cloned WIR15 gene, WTK1, encodes a tandem kinase-pseudokinase, conferring broad-spectrum resistance to stripast. The cloning of WIR15 paved the way to the discovery of a new protein family, termed here as tandem kinase-pseudokinases, TKPs. In a screening of more than 100 plant genomes, we have identified more than 3,000 TKPs. So those proteins are very common in the plant kingdom. And we find them from moss to trees, eucalyptus, wherever we looked for them, we found them. We propose a decoy role for the pseudokinase domain as a potential mechanism of function of the TKPs in the plant immune response. And these studies demonstrate the potential of wild MR gene pool for improvement of cultivated wheats by exploitation of genes that were lost or left behind during 10,000 years of domestication. I would like to thank my colleagues, and especially my former PhD students that conducted this research, like Valentina Klimyuk and Huang Ling and Andrei Fatyucha and Eli Tsuryaniv and Dina Ratz, which conducted this research, and my colleagues, Abraham Korol, Zeev Frankel, Ellen Schulman in Helsinki University, Morgan Hovmoller in Denmark, George Dubkovsky in UC Davis, Gita Quaker in Plant Pathology Department in UC Davis, Asaf Distelfeld at Tel Aviv University. Jaroslav Dolocel at Czech Republic, Thomas Wicker at University of Zurich, and Beat Keller and all the grant agencies. Thank you very much for your attention.